presenting on ether anesthetics. So before I get into ethers as general anesthetics, I would like to provide an introduction of what general anesthetics are. So what they are is basically compounds that generate the loss of responsiveness towards environmental stimuli for both humans and animals. And with the use of general anesthetics, it has led to a safe care in the medical environment, as well as big innovations in the medical field. And there have been many chemicals that have been used for anesthetics throughout history. However, there is one chemical that stands out, and that is the use of ethers. Ethers were the first anesthetic that was able to significantly do its job both safely and effectively. So in this presentation, I will explore the processes that have led to the discovery of ether as an anesthetic and the mechanism behind why ethers are able to work as an anesthesia. So the use of ethers as a anesthetic was discovered by a dentist from Boston named William T.J. Morton, and he was able to prove that ether in its gaseous state is able to be used effectively and safely as a general anesthetic when it is inhaled in a proper dose. And the type of ethers that are commonly used for anesthesia is usually diethyl ether, as you can see in the corner here on the bottom right. That is the general structure for diethyl ethers. And the use of diethyl ethers are still prevalent in low developing countries today because it has a very low cost and minimal risk in cardiac and respiratory depression. However, it has a high index of flammability, which is why it has led to less use in many developed countries. Before we can try to understand how ethers work specifically as a anesthesia, we should try to understand how most general anesthetics work. And when it comes to general anesthetics, the role of proteins is crucial in understanding and the mechanism of general anesthetics. And one of the functional proteins in our body include receptors that have a role in communicating cell signals. And these could be things such as proteins originating from hormones that induce changes in our body, as well as ion channels that have a role in controlling levels of ions such as sodium, potassium, and calcium, which is vital for brain function. And most of these regulating proteins tend to be spherical, and they usually have a hydrophobic cavity that is for accommodating other regulator molecules. And the binding of these regulator molecules to the regulating proteins usually activates a specific process or triggers an ion channel to open. And when an anesthetic molecule takes place of these regulator molecules, it ceases the normal activity and it usually leads to unconsciousness most of the time. This leads us to the question of whether there is a general anesthesia receptor. And the answer to this is yes, and it is the central cavity in all vital cell proteins. What does this mean? Well, these regulatory proteins and their corresponding regulatory molecules act like a biological lock and key, meaning that every regulatory protein has its own specific regulatory molecule that can fit into its cavity and no other molecule can take its place, kind of like a key and a lock. However, with an anesthetic molecule, it is able to occupy all locks, thus it prevents all keys from penetrating into their corresponding locks. And as you can see in the picture on the right, it's an example of an ion channel, and we have an anesthetic ketamine, which binds to it, and then it closes the ion channel, preventing it from establishing a gradient. So now we can move on to try to understand the mechanism of ethers, specifically as a general anesthetic. And around 170 years has passed since ethers was first used as a general anesthetic. However, the molecular mechanism of how ethers function as an anesthesia is still not completely understood to this present day, and it remains unclear. However, there have been studies that have been performed, and evidence have shown that ligand-gated ion channels and voltage-gated ion channels have been relevant targets for halogenated ethers when trying to induce unconsciousness. And generally, most general anesthesia also act through depression of excitatory signaling or enhancement of inhibitory signaling. And this generalization could lead us to many known proteins that could be prone to this, including glycine, glutamate, nicotinic acetylcholine, and many more. And on this figure on the right is a list of some of the halogenated ethers that have been used as general anesthesia. Alright, since voltage-gated ion channels could be a possible target for halogenated ethers, I'm going to get into the characteristics and mechanism of voltage-gated sodium channels. And what these are basically transmembrane protein complexes that are responsible for both the initiation and propagation of action potentials in both neurons and excitable cells. And these channels usually consist of a pore-forming alpha subunit as well as a non-pore-forming beta subunit, which tends to be smaller in size. And the alpha subunit usually plays a role in ion conduction as well as voltage sensing, while the beta subunits tend to have many roles, including modification of channel expression, gating, localization, and many more. And in 1940, a study by Heinbecker and Bartley was conducted where they reported an increase in the rate of accommodation to subthreshold depolarizations in voltage-gated sodium channels when a small amount of ether was introduced to it. From this result, they were able to conclude that the rate of accommodation is directly proportional to the rate of potassium channel activations as well as sodium channels inactivations. And this study was probably the first electrophysiological indication that ethers might have an effect on sodium channel function. And as you can see on the figure on the right, we have a diagram of what a voltage-gated sodium channel looks like. And the Roman numerals on the figure represent domains, while the numbers represent segments. And the pore loop segments are the ones in green, the ones in red are the voltage sensing segments, and the ones in yellow are the inactivation gate and the H particle. Now that we know about the potential correlation between ethers and voltage-gated sodium channels, we can then answer the question of what neurochemical consequences there might be when these channels are inhibited by these halogenated ethers. So in 2009, Hemmings proposed that there is a mechanism out there that where the presynaptic voltage-gated sodium channel inhibition would lead to the reduced depolarization of the presynaptic terminal. On top of that, the inhibition of these voltage-gated sodium channels would also lead to the reduction in calcium influx to the terminal using voltage-gated calcium channels. Then, this reduction in calcium would then lead to reduced exocytosis as well as reduced neurotransmission. Okay, so the mechanism of how ethers work as a general anesthesia still remains unclear until this day. However, studies have shown, as we saw earlier, that there are possible mechanisms of halogenated ethers on voltage-gated sodium channels, and these ethers are also known to be capable of targeting many proteins. It is therefore likely that these endpoints of the anesthetized state are likely to be attributed to different protein targets. And by understanding the role of voltage-gated sodium channels, it might just lead us to a further understanding of the mechanism of ethers as a general anesthetic. And although the use of ethers today is less prevalent as a general anesthetic, understanding its mechanism could help us make advances in anesthesiology, and perhaps the use of ethers might come back later on in the future once we are able to fully grasp of how it functions in the body and its molecular mechanism. Thank you for listening to my presentation. I hope you enjoyed and learned something today.